Hello there, and welcome back to Bedtime Science. I'm really glad you're here tonight. Tonight, we are going to rest inside one of the deepest questions human beings have ever asked: how life began. Not how animals appeared, not how forests grew, not how eyes, wings, flowers, or minds eventually evolved, but the quieter question underneath all of those later wonders. How did ordinary matter on a young, rocky planet cross the threshold into something that could use energy, keep itself together, and make imperfect copies of itself? The most peaceful way to approach this question is not to demand a single finished answer. The origin of life is still an open field of science. It sits at the meeting point of chemistry, geology, astronomy, physics, and biology. It asks about ancient oceans and falling meteorites, warm ponds and deep vents, RNA molecules and fatty membranes, minerals, energy gradients, and the first systems that could evolve. Some parts are known with confidence. Other parts are still being tested. That uncertainty is not a weakness. It is the honest shape of a question that reaches almost 4 billion years into the past. So tonight, we will move gently. We will not treat the origin of life as a hard wall to push against, but as a dark shoreline slowly becoming visible. One clue comes from ancient rocks. Another comes from every living cell. Another comes from laboratory chemistry. Another comes from meteorites and ocean worlds. Together, they do not yet make a final picture, but they do make something remarkable: a calm, evidence-based path toward understanding how chemistry became biology. Somewhere in the deep Archean, perhaps more than 3.7 billion years ago, and maybe earlier, on a young planet that had been bombarded by asteroids and was still geologically violent in ways the modern Earth is not, something happened whose consequences have never stopped unfolding. A collection of molecules crossed a threshold. From chemistry, biology emerged. From non-living matter, something began to grow, to consume energy, to maintain an internal state different from its surroundings, and eventually to make copies of itself. The young Earth is best imagined not as a quiet blue planet, but as a changing chemical world. Magma cooled into crust. Water condensed, boiled, and returned. Impacts disturbed the surface, while quieter pockets may have survived between violent intervals. The first living system did not need to appear fully formed. It only needed a setting where useful chemistry could persist longer than useless chemistry, and where repetition gave small advantages time to accumulate. We do not know exactly how it happened. We do not know where it happened. We do not know what the first self-replicating molecule looked like, or what the first protocell contained, or whether the transition from chemistry to biology was sudden or gradual, or somewhere in between. The origin of life is one of the deepest and most genuinely unsolved problems in science, a problem that sits at the intersection of chemistry, geology, physics, astronomy, and biology, and that every decade produces new experimental results, new theoretical frameworks, and new reasons to revise earlier certainties. The oldest traces of life are difficult because ancient rocks have been altered for billions of years. A carbon ratio may suggest biology, but it must be compared with non-biological chemistry. A tiny filament may look microbial, but minerals can imitate life. This uncertainty is not a flaw in the story. It is part of deep-time science. The farther back the evidence reaches, the more gently and carefully it has to be read. What we do know is astonishing. The ingredients were available. The conditions existed. The chemistry, as far as it has been reconstructed, can work. And whatever the specific pathway was, it succeeded once, or possibly more than once, but at least once, and from that beginning, for billion years of evolution produced everything alive today. The early ingredients were ordinary in one sense and extraordinary in another. Carbon, nitrogen, sulfur, phosphorus, metals, water, and energy are not alive by themselves. Yet together, in the right cycles and concentrations, they can become the raw grammar of biology. The wonder is not that one magical substance appeared, but that many simple substances could be arranged into systems that held together, reacted, and eventually remembered something about their own structure. Tonight we move through what science has learned and what it is still working out about the most consequential event in the history of this planet. Open science gives this topic its calm honesty. There are competing hypotheses, careful experiments, revised timelines, and arguments that continue because the evidence is difficult. A whole young planet supplied many natural laboratories at once: ponds, vents, shorelines, ice, minerals, volcanic gases, and falling meteorites. Some places would have destroyed fragile compounds. Others may have protected or concentrated them. The answer may be distributed across several environments rather than hidden in only one. The Earth formed approximately 4.54 billion years ago through the accretion of material in the solar nebula, the rotating disk of gas and dust that surrounded the young sun. The early Earth was a very different world from the one we inhabit. The surface was partially or fully molten in the early period, a magma ocean produced by the heat of accretion and by the radioactive decay of short-lived isotopes abundant in the early solar system. The atmosphere lacked free oxygen, which had not yet been produced in significant quantities by photosynthesis, and it probably changed through time, with carbon dioxide, nitrogen, water vapor, and smaller amounts of other gases playing important roles. Some local or temporary settings may have been more reducing than the planet's general atmosphere. The surface was subjected to intense bombardment by asteroids and comets during the late heavy bombardment, approximately 4.1 to 3.8 billion years ago, which repeatedly melted and vaporized any surface water that had accumulated and may have sterilized any early biosphere that had begun to develop. And yet the oldest evidence of life appears remarkably soon after the bombardment ended. Within a few 100 million years, microorganisms were present and metabolically active. The earliest possible environments matter because each one solves a different problem. Shorelines can concentrate molecules. Wet rocks can dry into reactive films. Ice can squeeze solutes into narrow channels. Clay can hold molecules in place. Vents can provide gradients and chemical energy. No setting has to solve everything alone. A plausible origin may have involved movement between environments, with molecules being formed in one place, concentrated in another, and reorganized somewhere else. The oldest physical evidence for life on Earth is disputed and actively debated in the scientific literature. Among the most widely discussed very ancient evidence are microfossil-like structures and carbon signatures preserved in rocks, such as the apex chert of the Pilbara region of Western Australia, where microfossils of filamentous microorganisms approximately 3.465 billion years old have been described. Though the biological origin of some of these structures has been challenged by researchers who argue they could be mineral formations that mimic microbial structures. Carbon isotope signatures in approximately 3.7 to 3.8 billion-year-old rocks from the Isua Greenstone Belt in Greenland have been interpreted as evidence of biological carbon fixation, based on the preferential incorporation of the lighter carbon-12 isotope over carbon-13 by photosynthetic and chemosynthetic organisms, a discrimination pattern preserved in the organic carbon of ancient rocks. Some researchers have proposed even older evidence from approximately 4.1 billion-year-old zircon crystals containing carbon inclusions with isotopic signatures consistent with biological origin, though this interpretation is extremely controversial. A cautious scientific view places the origin of life in the shadowy interval before the oldest widely discussed evidence for life, with some models pushing early cellular ancestry very far back, while the specific date remains uncertain. Common ancestry is one of the most peaceful facts in biology. The same deep molecular language appears across bacteria, archaea, plants, fungi, animals, and human cells. Ribosomes translate RNA. ATP carries energy. Nucleic acids store and transmit information. Membranes make an inside and an outside. These shared features point back toward Luca, not as the first life, but as the last ancestral cellular population whose descendants still exist today. All life on Earth shares a common ancestry, probably an ancestral population rather than a single lonely individual, from which all living cellular life descended through billions of years of evolution and diversification. This last universal common ancestor, abbreviated LUCA, is inferred from the features shared by all living organisms regardless of how distantly related they are. All cellular life uses DNA as its long-term genetic archive, encoded in the same 4 bases, while our RNA still carries messages and performs ancient catalytic work. All cellular life uses a nearly universal genetic code, the same correspondence between triplet DNA sequences and amino acids. All cellular life uses ribosomes to translate messenger RNA into protein. All cellular life uses ATP as a central energy currency and relies on deeply conserved molecular machinery, including ATP synthase, to manage chemical energy. Cellular life generally uses the same standard set of 20 amino acids. These universal features are most simply explained by descent from a common ancestor that had already developed all of these systems. They were inherited from LUCA rather than independently evolved multiple times. The remarkable universality of the molecular machinery of life is one of the most powerful lines of evidence that all life on Earth shares a single origin. RNA is attractive because it can sit between memory and action. It can carry sequence information, and some RNA molecules can catalyze reactions. That does not make the RNA world simple. RNA is chemically demanding, and a fully self-copying RNA system has not simply been found waiting in nature. But ribozymes, laboratory evolution, and nucleotide chemistry show why the idea remains powerful. RNA gives a possible bridge between chemistry that stores information and chemistry that performs work. The RNA world hypothesis is one of the most widely discussed theoretical frameworks for understanding how genetic information and biochemical catalysis were first integrated into a self-replicating system. The central challenge in understanding life's origin is the chicken and egg problem of modern biochemistry. DNA stores genetic information but cannot catalyze chemical reactions. Proteins catalyze reactions but cannot store genetic information, and the production of each requires the other. How could this interdependent system have originated? RNA provides a possible resolution because RNA molecules can both carry genetic information, like DNA, and catalyze chemical reactions, like proteins. RNA molecules called ribozymes that catalyze specific chemical reactions, including reactions involved in RNA replication, have been discovered in living cells and produced in the laboratory. The hypothesis proposes that RNA was the original information storage and catalytic molecule, predating both DNA and proteins, and that DNA and protein later evolved as more efficient specialists for information storage and catalysis respectively, with RNA retaining both functions in a diminished capacity in modern cells. Prebiotic chemistry is quieter than the word synthesis can make it sound. It is not a factory line. It is light, minerals, temperature, acidity, drying, wetting, and chance encounters among small molecules. Cyanide chemistry, sulfur chemistry, phosphate chemistry, and ultraviolet light can all be useful or destructive depending on context. The same environment that makes one molecule may break another. Origins research is partly the search for sequences of conditions that help more than they harm. The spontaneous formation of RNA on the early Earth presents significant chemical challenges that have been the subject of intensive research. The nucleotides that compose our Na, each consisting of a nucleobase, a ribose sugar, and a phosphate group, must be assembled in the correct sequence, and the polymerization of nucleotides into our Na chains requires removing water molecules at each step, a reaction that does not occur spontaneously in aqueous solution under ordinary conditions. Research by John Sutherland and his group at the MRC Laboratory of Molecular Biology in Cambridge has demonstrated elegant chemical pathways that can produce our Na nucleotides from simple prebiotic precursors, hydrogen cyanide, hydrogen sulfide, water, and ultraviolet light, in reaction sequences that occur under plausibly prebiotic conditions. This work helped soften what had previously seemed like a nearly insurmountable chemical obstacle and provided the strongest experimental support yet for the chemical plausibility of our Na world origins. The key insight was that attempting to assemble our Na from its component parts, nucleobase plus sugar plus phosphate, was chemically difficult, but building the components simultaneously through a shared chemical pathway was considerably more tractable. Hydrothermal vents offer a picture of life beginning from flow. Alkaline fluids rich in hydrogen meet a more acidic ocean. Minerals create tiny pores. Carbon dioxide, metals, and redox gradients are present together. In such a setting, energy is not a separate ingredient added later. It is built into the landscape. The appeal of the vent hypothesis is that it gives early chemistry a continuous engine, a way for reactions to be pushed forward by the planet itself. Hydrothermal vents are one of the most actively studied proposed environments for the origin of life, particularly the alkaline hydrothermal vents of the type represented by the Lost City vent field in the mid-Atlantic. The Lost City vents, discovered in 2000, are driven not by volcanic heat, but by serpentinization, the chemical reaction between seawater and the iron and magnesium-rich mantle rock peridotite, which generates heat and produces highly alkaline, hydrogen-rich fluid. The hypothesis of alkaline vent origin, developed primarily by Michael Russell and William Martin, proposes that the natural pH gradient across the thin mineral walls of ancient submarine vents, alkaline inside the vent, more acidic in the ancient ocean, could have driven proton flow across these inorganic membranes in a way that is analogous to the proton gradient that powers ATP synthesis in modern cells. On this model, the first proto-metabolic reactions were driven not by light, but by the geochemical energy of the hydrogen-rich vent fluid reacting with carbon dioxide in the acidic ancient ocean, and the mineral pores of the vent structure provided the compartmentalization that allowed concentration of reactants and products. Warm ponds and terrestrial hydrothermal fields offer a different gift: cycles. A shallow pool can fill with water, concentrate as it evaporates, dry into a film, and refill again. Each cycle changes the chemistry. Molecules that were too dilute may become crowded. Reactions that were unlikely in open water may become possible on a damp mineral surface. The surface world also allows ultraviolet light to drive some useful reactions, even as it threatens fragile products. Darwin's warm little pond, a suggestion made in a private letter of 1871 in which Darwin speculated that life might have originated in a warm pond with the right chemical conditions, has been revived as a relevant environment for life's origin in the context of terrestrial hydrothermal fields. The work of Nick Lane, John Allen, and others has highlighted that shallow, periodically wet, dry hydrothermal environments on land, like those found today at Yellowstone or the geothermal regions of Iceland and New Zealand, offer conditions favorable for the concentration and polymerization of organic molecules. The wet-dry cycling that occurs as pools alternately fill with water and dry up in the heat can concentrate organic molecules and drive polymerization reactions that do not occur in dilute aqueous solution, potentially providing a mechanism for the formation of long RNA polymers and lipid membranes. The periodic flooding with fresh water from rain or runoff provides repeated opportunities for new combinations of molecules, and the ultraviolet light available at the surface, while potentially damaging to complex organic molecules, can also drive the kind of photochemistry that produces nucleotide precursors through the Sutherland pathway. Membranes make the origin of life feel more physical. A fatty acid vesicle is simple compared with a modern cell, but it already creates an inside and an outside. Molecules trapped inside are no longer lost immediately to the larger world. If those molecules help the vesicle grow or divide, the boundary and the contents begin to share a fate. This shared fate is one of the quiet beginnings of individuality. Lipid membranes, the thin, self-assembling sheets of amphiphilic molecules that enclose the contents of all living cells, are essential for any protocell because they provide the compartmentalization needed to keep a self-replicating system together, maintain concentration gradients, and separate the internal chemistry from the external environment. The research of Jack Szostak at Harvard Medical School demonstrated that fatty acids, simpler lipid molecules than the phospholipids of modern cell membranes, can spontaneously assemble into vesicles under plausible prebiotic conditions, and that these vesicles can grow, divide, and encapsulate RNA molecules. Fatty acid vesicles can grow by absorbing additional fatty acid molecules from their surroundings, and they can divide into daughter vesicles when agitated, a simple protocell division that does not require any protein machinery. Vesicles containing RNA can selectively grow at the expense of empty vesicles because the osmotic pressure of the RNA inside drives water into the vesicle, increasing its surface area and enabling preferential incorporation of additional fatty acids. This simple selective advantage of RNA containing over empty vesicles provides a primitive selection pressure that could have driven the evolution of the earliest protocells. Protocell evolution is powerful because it links several small advantages. A vesicle that contains useful RNA may draw in water, grow faster, divide more often, or keep helpful molecules together. None of this requires intention. It only requires variation and consequences. Empty vesicles, leaky vesicles, crowded vesicles, and chemically active vesicles would not all behave the same. Once some compartments leave more descendants than others, selection has a place to begin. The Miller-Urey experiment, conducted in 1953 by Stanley Miller under the supervision of Harold Urey at the University of Chicago, was the first experimental demonstration that organic molecules relevant to life could be produced from simple inorganic precursors under conditions simulating the early Earth. Miller passed an electrical discharge, simulating lightning, through a mixture of water, methane, ammonia, and hydrogen, thought at the time to represent the reducing atmosphere of the early Earth. After 1 week, the water in the apparatus had turned pink and then reddish-brown. An analysis of the contents revealed that amino acids, including glycine and alanine, the simplest of the 20 amino acids used in proteins, had been produced spontaneously. The experiment was a landmark because it demonstrated for the first time that organic chemistry could bridge the gap between simple inorganic molecules and the building blocks of life. Reanalysis of Miller's original samples, preserved after his death and studied with modern analytical techniques in 2008, revealed an even richer mixture of amino acids than originally detected, including several not found in natural proteins, confirming that the experiment was even more productive than the original analysis had suggested. The Miller-Urey experiment remains important not because it perfectly reproduced the early Earth, but because it changed the imagination of science. It showed that biologically relevant organic molecules could arise from simple gases and energy. Later work made the picture more cautious, because the early atmosphere was probably not the strongly reducing mixture Miller used. But the deeper lesson survived. Non-living chemistry can produce many of life's building blocks under plausible natural conditions. The composition of the early Earth's atmosphere is now understood to have been different from what Miller and Urey assumed. The original experiment used a highly reducing mixture of methane and ammonia, but geological evidence now suggests that the early atmosphere was less reducing, dominated by carbon dioxide and nitrogen, with relatively little methane or hydrogen. A less reducing atmosphere produces organic molecules less efficiently in Miller-Urey type experiments, though it does not prevent their formation entirely. Subsequent researchers have explored a range of atmospheric compositions and energy sources: ultraviolet light, hydrothermal chemistry, impact shock chemistry, and found that organic molecules including amino acids, nucleotide bases, and sugars can be produced under a wide range of conditions. The accumulation of organic molecules on the early Earth was probably supplemented by delivery from space. Carbonaceous chondrite meteorites contain amino acids, nucleobases, and many other organic molecules that formed in the early solar system, and the delivery of such material may have been an important addition to chemistry already happening on Earth. Meteorites add the solar system to the story. Carbonaceous chondrites contain amino acids, nucleobase-related compounds, sugars, and many other organic molecules. The Murchison meteorite is famous because it revealed a rich extraterrestrial organic inventory, much of it not produced by biology. This does not mean life arrived ready-made from space. It means Earth was not chemically isolated. The young planet received ingredients from its wider neighborhood, while its own chemistry was also active. The Murchison meteorite, which fell in Australia in September 1969, is one of the most thoroughly studied meteorites in history and has provided a wealth of information about the organic chemistry of the early solar system. Over 7 decades of analysis, researchers have identified more than 70 different amino acids in the Murchison meteorite, including many amino acids used by biology, as well as many others not normally used in proteins, along with nucleobase-related compounds, sugars including ribose reported in carbonaceous meteorites, lipid-like compounds, and a vast array of other organic molecules. The total number of distinct organic compounds identified in the Murchison meteorite now exceeds 14,000, and the complexity of the extraterrestrial organic chemistry revealed is remarkable. The amino acids in the Murchison meteorite are nearly racemic, nearly equal mixtures of left-handed and right-handed forms. Unlike the exclusively left-handed amino acids of biology, though a slight excess of left-handed amino acids has been detected in some amino acid classes, possibly related to the origin of life's homochirality. Handedness gives early life a subtle problem. Biology uses mostly left-handed amino acids and right-handed sugars, creating polymers that fold in consistent ways. Ordinary chemistry often makes both hands together. A tiny initial bias may have come from chance, minerals, polarized light, or extraterrestrial chemistry, but that bias then had to be amplified. Homochirality is a reminder that life is not only about which molecules exist, but about the ordered way they are used. Homochirality, the exclusive use of left-handed amino acids and right-handed sugars in all living things, is one of the more puzzling features of biochemistry from the perspective of life's origin. Most chemical processes that produce chiral molecules produce racemic mixtures of both handedness equally. Yet all proteins on Earth are made exclusively of left-handed amino acids, and all nucleic acids use right-handed sugars. The functional reason for this uniformity is clear. A polymer chain of mixed handedness would not fold into a stable structure, but the historical reason for why one-handedness was selected over the other is not established. Proposed explanations include an initial random symmetry breaking event amplified by autocatalytic reactions, polarization effects from cosmic rays or polarized light in the early solar nebula, and the possibility of a small initial excess of one-handedness in the prebiotic organic inventory delivered by meteorites being amplified by subsequent chemistry. The origin of homochirality remains one of the open questions in the study of life's origin. Translation may be the hardest bridge to imagine because it connects two languages. Nucleotide sequences must correspond to amino acid sequences. Codons must meet transfer RNAs. Ribosomes must link amino acids into proteins. The modern system is highly refined, but the ribosome's catalytic heart is our RNA, which suggests an ancient route from an RNA-rich world toward a protein-rich one. Protein synthesis may have begun as something smaller, rougher, and less exact before becoming universal. The transition from a world of self-replicating RNA to the modern cellular world in which DNA stores information, proteins do chemistry, and RNA serves as intermediary required a series of evolutionary innovations whose order and mechanism remain active research questions. The evolution of the genetic code, the correspondence between RNA triplets and amino acids that allows ribosomes to translate genetic information into protein structure, is one of the most central puzzles. The code is essentially universal across all life, and its specific assignments of codons to amino acids show patterns suggesting that it evolved gradually, with the code beginning with a small number of amino acid codon assignments and expanding over time. The evolution of the ribosome itself, the molecular machine that translates RNA into protein, was a prerequisite for the protein world that eventually displaced the RNA world, and the ribosome retains clear signs of its RNA-based origin in the fact that its catalytic activity resides in RNA rather than protein. The ribosome is, in a meaningful sense, a fossil of the RNA world, a molecular relic of the earliest period of biological evolution preserved in modified form in every living cell. Early gene sharing makes the tree of life less like a single clean trunk. In young cellular communities, genetic material may have moved frequently among lineages. Boundaries between groups could have been loose. Useful innovations may have spread sideways, not only downward from parent to offspring. This communal view does not erase common ancestry. It makes early ancestry more like a braided river, with channels splitting, rejoining, and slowly becoming more defined. Horizontal gene transfer, the movement of genetic material between organisms other than through parent to offspring inheritance, was probably far more extensive in the early biosphere than it is in the modern world, where it remains especially important among bacteria and archaea. In the early period of cellular evolution, before the three domains of life, bacteria, archaea, and eukarya, had diverged sufficiently to develop the molecular incompatibilities that now largely prevent gene transfer between domains. The exchange of genetic material between protocellular lineages may have been so extensive that the concept of distinct evolutionary lineages is not meaningful. The last universal common ancestor, Luca, may not have been a single organism, but a population of organisms exchanging genes so frequently that they formed a gene pool rather than a genealogical tree, with the three domains of life emerging as gene exchange frequencies declined and distinct cellular lineages became established. This view, associated with Carl Woese and others, suggests that the earliest stage of cellular evolution was fundamentally communal in nature. Extremophiles widened our sense of what life can tolerate. Heat, pressure, salt, acid, radiation, cold, and darkness are not automatic barriers. Modern extremophiles are not living fossils from the origin of life, because they too have evolved for billions of years. Still, They show that biology can adapt to conditions once considered impossible. That matters for early Earth, and it also matters for ocean worlds and ancient Mars. The discovery of extremophiles, organisms that live under conditions previously thought to be incompatible with life, has dramatically expanded the known range of environments where life is possible and has implications for understanding the conditions under which life first emerged. Thermophilic bacteria and archaea live in hot springs at temperatures up to approximately 80 degrees Celsius. Hyperthermophilic archaea live in hydrothermal vents at temperatures up to approximately 121 degrees Celsius, well above the normal boiling point of water at the surface, but liquid at the pressures of the deep ocean floor. Acidophiles live in environments with pH as low as 0, the extreme acidity of certain mine drainage waters. Alkalophiles live at pH values up to 11 or higher. Halophiles thrive in saturated salt solutions. Psychrophiles are metabolically active at temperatures well below freezing in the brine channels of sea ice. Piezo live under pressures thousands of times atmospheric. Radiation-resistant organisms like Deinococcus radiodurans can survive ionizing radiation doses that would kill other organisms by orders of magnitude. Each discovery of life in a new extreme environment extends the range of conditions that might support life and suggests that the origin of life could have occurred under conditions far outside the mild, temperate range that was originally assumed. Panspermia has to be handled carefully. Organic molecules from space are real and important. Rocks can move between planets, and Martian meteorites have reached Earth. Some microbes or spores can survive harsh conditions for limited periods in experiments. But none of this proves that life came from elsewhere. Chemical delivery is well supported. The delivery of living organisms remains uncertain. Directed panspermia has no evidence and does not help explain how life began in the first place. The panspermia hypothesis, the proposal that life or the precursors of life were delivered to Earth from space, exists in several versions of varying scientific credibility. Lithopanspermia, the most scientifically plausible version, proposes that microbial life could survive transport between planetary bodies on fragments of rock ejected by asteroid impacts, protected from radiation and vacuum by the rock itself. There is genuine evidence that such transport is possible. Martian meteorites have been found on Earth, and modeling of impact ejecta shows that material from Mars can reach Earth on trajectories consistent with lithopanspermia. Organisms, including bacterial spores, have been found to survive simulated space conditions for extended periods in laboratory experiments. Whether any organism could survive the full duration of interplanetary transit, potentially thousands of years, and the heat of atmospheric entry is uncertain but not definitively ruled out. Directed panspermia, the deliberate seeding of Earth by intelligent extraterrestrial life, has no evidence in its support and sits outside the evidence-based core of origin of life research. Chemical panspermia, the delivery of organic molecules from space, as discussed in the context of carbonaceous meteorites, is well established and is considered an important supplement to terrestrial organic chemistry. Photosynthesis changed the planet long after life's origin. Early photosynthesis probably did not release oxygen. Oxygenic photosynthesis, later developed by cyanobacteria, split water and released oxygen as waste. Over time, that oxygen transformed oceans, atmosphere, minerals, and metabolism. The Great Oxidation Event was not the first chapter of life, but it was one of the great turning points. It shows how microscopic cells can eventually rewrite a whole planet. The origin of photosynthesis, the ability to use light energy to fix carbon dioxide into organic molecules, was one of the most consequential innovations in the history of life. Anoxygenic photosynthesis, which does not produce oxygen as a byproduct, appears to have evolved first, in bacteria that used hydrogen sulfide or other electron donors rather than water. Oxygenic photosynthesis, which splits water to extract electrons and releases oxygen as a byproduct, evolved later in the cyanobacteria and is responsible for the oxygenation of Earth's atmosphere. Oxygenic photosynthesis requires two photosystems working in series, which is mechanistically more complex than the single photosystem of anoxygenic photosynthesis, and the evolutionary transition between them is an active research question. The oxygen produced by cyanobacterial photosynthesis accumulated in the atmosphere, beginning approximately 2.4 billion years ago in the Great Oxidation Event, transforming the atmospheric chemistry of the planet and making possible the evolution of aerobic respiration, a metabolic strategy that can extract far more usable energy from glucose than fermentation alone, and that provided the energetic foundation for the evolution of complex multicellular life. Eukaryotic cells show how cooperation can become structure. Mitochondria were once bacteria that became permanent partners inside another cell. Chloroplasts came later from cyanobacteria and the ancestors of plants and algae. These events did not explain the first life, but they show a pattern repeated in evolution. Life becomes more complex not only by inventing new parts, but by bringing existing living systems into new relationships. The endosymbiotic origin of the eukaryotic cell was one of the great later thresholds in the history of life, long after the origin of the first self-replicating systems. Eukaryotic cells, those with membrane-bound nuclei and complex internal organization, arose approximately 1.5 to 2 billion years ago from the merger of an archaeal host cell with one or more bacterial endosymbionts. The mitochondria of all eukaryotic cells are the descendants of alpha proteobacteria engulfed by the ancestral archaeal cell in a single endosymbiotic event, and the chloroplasts of photosynthetic eukaryotes are the descendants of cyanobacteria engulfed by a eukaryotic ancestor in a subsequent endosymbiotic event. The evidence for these endosymbiotic origins is overwhelming. Mitochondria and chloroplasts retain their own circular DNA, their own ribosomes of bacterial type, and their own protein synthesis machinery, and phylogenetic analysis places them firmly within the bacteria. The fusion of the information processing capabilities of the archaeal host, including the complex gene regulation and nuclear organization of archaea, with the energy-generating capabilities of the bacterial endosymbiont, may have been the key event that enabled the evolution of the complex, energy-intensive eukaryotic cell body plan. Protocells make the abstract question visible. Imagine a small membrane sphere in water, not alive in the modern sense, but able to grow, divide, and keep some molecules close together. If the chemistry inside helps the sphere continue, and the sphere helps the chemistry persist, the parts begin to form a system. The threshold may have looked like this: not a sudden cell, but a gradually tightening relationship between boundary, information, and energy. The RNA world, if it existed as proposed, was not a single moment, but a long evolutionary period during which the molecular machinery of life was being elaborated. The earliest RNA molecules capable of self-replication would have been simple and error-prone, with high mutation rates producing extraordinary diversity. Natural selection acting on this diversity would have favored RNA molecules that replicated more accurately, that could catalyze useful reactions, that formed cooperative networks with other RNA molecules, and eventually that could be encapsulated in lipid vesicles. The transition from free-floating RNA to RNA enclosed in vesicles was a threshold moment. The first protocell, because encapsulation linked the fate of the RNA to the fate of the vesicle, creating a selective unit at the level of the protocell rather than the individual molecule. Protocells that were better at replicating their RNA content would produce more daughter vesicles carrying the same RNA, and the competition between protocells for fatty acids and nucleotides would have driven the evolution of more efficient protocells. The genetic code is nearly universal, and that universality is one of the strongest signs of shared ancestry. Its organization also looks non-random. Similar amino acids often correspond to related codons, which may reduce the harm caused by some errors. Whether the first assignments came from chemical affinities, historical accidents, selection for robustness, or all of these together, the code became so central that later life could hardly change it without changing everything. The genetic code, the correspondence between triplets of RNA bases, called codons, and specific amino acids, is essentially universal across all life on Earth, with only very minor variations in a few organisms. The near universality of the code is powerful evidence for a single origin of the translation machinery. If the code had evolved independently in different lineages would be expected to use different code tables. The specific assignments of codons to amino acids in the universal genetic code appear to be non-random. Amino acids with similar chemical properties tend to have similar codons, and the code is organized in ways that minimize the deleterious effects of mutations and translation errors. This non-randomness suggests that the code evolved through selection for robustness rather than being simply frozen in its initial form. The stereochemical hypothesis for the origin of the code proposes that the initial codon amino acid assignments were based on direct physical chemical affinities between specific RNA triplets and specific amino acids, an idea that has received some experimental support through studies of aptamers, short RNA sequences selected for binding to specific amino acids. The microbial age gives the story its deepest perspective. For most of Earth's history, life was microscopic. The machinery that later supported animals, plants, and complex ecosystems was being developed in cells too small to see without instruments. Metabolic pathways, membranes, genes, and regulatory systems accumulated long before the Cambrian explosion. The origin of life is therefore not a quick preface to animals. It is the opening of a long microbial planet. The Cambrian explosion, approximately 541 to 520 million years ago, was the period during which most of the major animal body plans appeared in the fossil record, an event that occurred approximately 3 billion years after the first life appeared. In the broad context of life's history, the first 3 billion years were dominated by microbial life, and animals are a very recent addition to the biosphere. The remarkable speed of animal diversification in the Cambrian relative to the preceding 3 billion years of microbial evolution reflects multiple factors. The evolution of complex developmental genetics encoding instructions for building complex body plans, the rise of predation creating new selective pressures and ecological interactions, the increase in atmospheric oxygen to levels sufficient to support the metabolic demands of active predatory animals, and possible changes in ocean chemistry that provided new resources and ecological opportunities. But the raw material for this diversification, the genes, the metabolic pathways, the cellular machinery, had been accumulating in single-celled organisms for 3 billion years before the animals appeared. Cosmic chemistry connects life's beginning to the wider universe. Carbon compounds exist in interstellar clouds. Meteorites contain organic molecules. Stars created the heavier elements that chemistry later used. Helium is abundant but mostly chemically silent, while carbon is versatile enough to build chains, rings, and complex frameworks. Earthly life is carbon-based not by accident alone, but because carbon offers a vast chemical vocabulary under ordinary planetary conditions. The deep connections between the chemistry of life and the chemistry of the cosmos are one of the most philosophically significant aspects of the origin of life problem. The most abundant elements in the universe, hydrogen, helium, oxygen, carbon, nitrogen, are also the most abundant elements in living organisms, with helium being the exception because it does not form chemical bonds. The organic molecules found in interstellar molecular clouds, hydrogen cyanide, formaldehyde, glycolaldehyde, ethylene glycol, and dozens of others, are the same molecules that serve as the starting materials for the prebiotic chemistry of life's building blocks. The carbon-based chemistry of life is also the most chemically rich chemistry accessible in the cosmos. Carbon forms for covalent bonds, can bond to itself in chains and rings of unlimited length and complexity, and its compounds with hydrogen, oxygen, nitrogen, and phosphorus encompass a combinatorial space of molecular structures that dwarfs that available to any other element. Whether life elsewhere in the universe would also be carbon-based remains an open question, but the abundance of carbon throughout the cosmos and its unique chemical versatility make it a strongly favored candidate. Probability remains an honest mystery. Life appeared early on Earth, but Earth is the only inhabited planet we know. Some scientists read the quick appearance of life as a sign that life may be likely when conditions are right. Others warn that we can only observe from a planet where life did appear, so the example is biased. The data are still too thin for certainty. The question remains open, and that openness is part of its power. The question of whether the origin of life was a unique, highly improbable event, occurring perhaps only once in the universe, or an inevitable outcome of the chemistry available under the conditions that prevailed on the early Earth, is one of the deepest unresolved questions in astrobiology. If life's origin required an extraordinarily improbable sequence of chemical events, then life might be vanishingly rare in the universe despite its existence on Earth. If life's origin is a likely consequence of the kind of chemistry available on rocky planets with liquid water, usable energy, and the right local chemical conditions, then life may be widespread in the cosmos. The speed with which life appeared after the conditions on Earth became suitable, within approximately a few 100 million years of the formation of stable oceans, suggests to some researchers that life's origin is relatively probable given the right conditions, since a long waiting time for an improbable event would predict a longer delay between habitability and the appearance of life. Others argue that we are necessarily on a planet where life appeared quickly, the anthropic principle, and that the speed of life's appearance on Earth tells us nothing about how difficult the origin of life is in general. Biosignatures extend the origin question into the sky. Oxygen with methane, chemical disequilibrium, seasonal patterns, and unexpected atmospheric combinations may suggest biology, but context matters. A gas can have several sources. A world can mislead if only one measurement is considered. The search for life beyond Earth is therefore slow and careful. It asks not just whether a molecule is present, but whether the whole planet makes sense as a living system. The detection of life beyond Earth, or the confirmation of its absence, would be the most consequential scientific discovery in human history. The search for biosignatures, chemical or physical indicators of biological processes, in the atmospheres of exoplanets is one of the primary scientific goals of current and planned space observatories. The simultaneous presence of oxygen and methane in an exoplanet atmosphere would be a strong biosignature because the two gases react chemically and would quickly disappear in the absence of continuous biological production of at least one of them. The presence of complex organic molecules, unexpected chemical disequilibria, or seasonal variations in atmospheric composition consistent with biological cycles are other proposed biosignatures. Within the solar system, the search for life focuses on Mars, where evidence of ancient habitable conditions is strong, and on the subsurface oceans of Europa, Enceladus, Titan, and Ganymede. Future missions to Europa and Enceladus, specifically targeting the detection of biosignatures in water plumes or subsurface ocean samples, are among the highest scientific priorities in planetary science. The transition from chemistry to biology can be described as the arrival of heredity, variation, and selection. A reaction network may be complex, but without heritable memory, it cannot evolve in the Darwinian sense. A copying molecule may carry memory, but without energy and materials, it cannot continue. Life begins to look plausible when these pieces overlap. Copying, boundaries, metabolism, and differences that affect survival. The study of the origin of life is ultimately a study of the transition from the universe described by chemistry and physics to the universe that includes biology, from a cosmos in which complex molecules interact through the ordinary laws of thermodynamics and kinetics to one in which some of those molecules have organized themselves into systems capable of heredity, variation, and selection. This transition, wherever it occurred and however it occurred, changed the character of a planet in ways that nothing else has. The atmosphere was transformed. The surface was transformed. The cycling of elements through rock, ocean, and air was transformed. And eventually, after nearly 4 billion years of biological evolution, the transformation produced organisms that became curious about how it all began. Ribozymes keep the RNA world from being merely an idea. Their discovery showed that RNA can catalyze reactions, not only carry information. The ribosome itself preserves this ancient clue because the central reaction of protein synthesis is catalyzed by RNA. Modern cells are full of proteins, but at their core is a memory of an older chemistry. The past is not gone. It is embedded inside the machinery still making proteins now. The answer is not fully known. The work continues. Chemical evolution came before biological evolution in the strict sense. Molecules could be favored because they were more stable, more reactive in useful ways, better at binding to surfaces, or more likely to be incorporated into growing networks. This was not natural selection as cells experience it, but it could prepare the ground. Before genes competed, molecular systems may have persisted or disappeared according to their chemistry. The study of ribozymes, RNA molecules that catalyze chemical reactions, has been central to building the experimental case for the RNA world hypothesis. The discovery of ribozymes by Thomas Cech and Sidney Altman in the early 1980s, for which they shared the Nobel Prize in Chemistry in 1989, demonstrated for the first time that RNA could function as an enzyme. Before this discovery, all known biological catalysts were proteins, and the chicken and egg problem of how proteins could be synthesized without the protein machinery needed to make them seemed intractable. Ribozymes provided the escape. RNA could both carry information and catalyze reactions, potentially bootstrapping itself into existence without any protein assistance. Since Cech and Altman's discovery, many more ribozymes have been found in living cells, performing essential cellular functions including the splicing of pre-messenger RNA, the synthesis of transfer RNA, and the peptidyl transfer reaction at the core of ribosome function. The very reaction that synthesizes proteins is catalyzed by RNA rather than protein. A living fossil of the RNA world preserved at the heart of the modern cell. Nitrogen is essential but stubborn. Molecular nitrogen is abundant in the atmosphere, yet its triple bond makes it hard to use. Lightning, impacts, volcanic chemistry, and meteorites could have supplied reactive nitrogen before biological nitrogen fixation evolved. Later, nitrogenase allowed some microbes to pull nitrogen from the air directly. That enzyme's oxygen sensitivity fits the deep history of life before oxygen filled the atmosphere. The concept of chemical evolution, the gradual increase in molecular complexity through chemical selection before the emergence of Darwinian biological evolution, was introduced by the British biologist John Haldane in 1929 and elaborated by Alexander Oparin in his 1938 book, The Origin of Life. Oparin proposed that the early ocean was a primordial soup rich in organic molecules, and that the spontaneous formation of droplets, coacervates, in Oparin's terminology, provided the compartmentalization needed for the evolution of more complex chemistry within a defined boundary. While the specific details of Oparin's model have been superseded by subsequent research, the core idea that chemical evolution in a compartmentalized environment preceded Darwinian biological evolution remains central to most contemporary models of life's origin. The distinction between chemical evolution, which is driven by the thermodynamic and kinetic properties of molecules rather than by heredity and natural selection, and biological evolution, which requires heritable variation, is an important conceptual boundary at which the origin of life is located. Energy may be the quietest necessity. A molecule cannot keep organizing itself without a flow of usable energy. Modern cells use ATP and proton gradients, but early systems needed simpler beginnings. Vents could provide natural proton gradients. Redox reactions could move electrons. Phosphate compounds, or thioesters, could help drive difficult reactions. The first energy systems were probably rough, local, and inefficient, but good enough to keep chemistry from sliding back into sameness. The nitrogen cycle is fundamental to understanding life's origin because nitrogen is an essential component of amino acids and nucleotides, the building blocks of proteins and nucleic acids. Molecular nitrogen is the dominant form of nitrogen on Earth and in most environments, but it is chemically inert and cannot be used directly by most organisms. The fixation of nitrogen, its conversion from molecular nitrogen to ammonia or other biologically available forms, is therefore essential for biology. On the early Earth, nitrogen fixation occurred through lightning discharges, which break the nitrogen-nitrogen triple bond and allow nitrogen to combine with oxygen or hydrogen to form reactive nitrogen compounds. The delivery of fixed nitrogen from meteorites also contributed. The eventual evolution of biological nitrogen fixation, the ability to fix atmospheric nitrogen directly using the enzyme nitrogenase, found in certain bacteria and archaea, was an important biological innovation that made nitrogen fixation independent of geochemical processes. The active site of nitrogenase contains an iron-molybdenum cofactor of unusual structure, and the enzyme is extraordinarily sensitive to oxygen, suggesting it evolved in an anoxic environment, consistent with an origin before the Great Oxidation Event. Minerals may have served as early scaffolds. Clay surfaces can concentrate molecules and help line them up. Metal sulfides can participate in electron transfer. Pores can trap small mixtures. Surfaces can protect some molecules and expose others to reaction. A mineral is not alive, but it can shape chemistry. In the origin of life, rock may have acted like a patient, silent partner before cells learned to build their own catalysts. The problem of energy for the first living systems is one of the central challenges in understanding the origin of life. Modern cells use ATP as their primary energy currency, and ATP is produced by ATP synthase using the proton gradients generated by the oxidation of organic molecules or by photosynthesis. The origin of this energy system, which requires sophisticated protein machinery, presents a chicken and egg problem similar to that of the RNA world. How could the first cells produce energy without the protein machinery needed to produce energy? The alkaline vent hypothesis addresses this by proposing that the first proto-metabolic reactions were driven by natural proton gradients across the thin mineral walls of submarine vent chimneys, providing energy in a form compatible with what ATP synthase later evolved to exploit. Other hypotheses focus on pyrophosphite and other simple phosphate compounds as primitive energy currencies that could be produced abiotically and used to drive biosynthetic reactions before the evolution of ATP. The specific mechanism of energy generation in the first living systems remains an open question. The Darwinian threshold is the moment when chemistry begins to have descendants in a meaningful way. Copies are made. Copies differ. Some differences affect the likelihood of being copied again. Once that loop exists, time changes character. It is no longer only time passing over reactions. It is time accumulating history. From that threshold onward, the past can influence the future through heredity. The role of mineral surfaces in the origin of life has been studied extensively since the 1980s. Clay minerals such as montmorillonite have been shown to catalyze the polymerization of amino acids and nucleotides into longer chains under prebiotic conditions. The regular, charged surfaces of clay minerals can orient and concentrate nucleotides, lowering the energy barrier for polymerization. Jack Ferris and colleagues demonstrated that activated nucleotides, produced through reactions with simple condensing agents, could be polymerized by montmorillonite into RNA chains up to approximately 50 nucleotides in length, long enough to fold into structured RNA molecules with potential catalytic activity. Iron-sulfur minerals such as pyrite have been proposed by Gunter Wachter-Schauser as the catalytic surface on which the first biosynthetic reactions occurred, with the reducing power of the pyrite surface driving the fixation of carbon dioxide into organic molecules. Metal sulfides are the active centers of many ancient and fundamental enzymes, including those involved in nitrogen fixation and the citric acid cycle, suggesting that metal sulfide chemistry was central to early biochemistry and may have been inherited from prebiotic mineral catalysis. The standard set of 20 amino acids is not the only possible set. Many other amino acids exist, and some appear in meteorites or in non-standard biological roles. The 20 used in proteins cover a useful range of sizes, charges, shapes, and chemical behaviors. Once the genetic code became fixed around them, changing the set became difficult. History hardened into biology, and biology carried that history forward. The emergence of Darwinian evolution from pre-biological chemistry required the development of a hereditary system in which information could be copied and in which variants of the information differed in their ability to replicate. The minimal requirements for a Darwinian system are usually stated as multiplication, the ability to make copies, heredity, the ability to pass information from parent to offspring, and variation, the existence of heritable differences among individuals. Once these conditions are met, natural selection will act on the variation and evolution will occur. The challenge in the origin of life is explaining how multiplication and heredity first arose, how the first informational molecules began to copy themselves with sufficient fidelity that a population of variants could be maintained and selected upon. Laboratory evolution experiments using RNA molecules, starting with random sequence RNA pools and selecting for molecules with specific biochemical activities, have demonstrated that RNA populations can evolve in vitro and have produced ribozymes with activities not found in nature, including RNA polymerase ribozymes capable of copying short RNA templates. These experiments provide proof of principle for the RNA world and illuminate the early stages of molecular evolution. Defining life remains difficult because the transition was probably gradual. A membrane vesicle is not enough. A replicating molecule may not be enough. A metabolic network may not be enough. But when these features become linked, a new kind of system appears, one that maintains itself, copies information, varies, and can evolve. The boundary between non-life and life may not have been a line, but a widening dawn. The question of why life uses exactly 20 amino acids, neither more nor fewer, has been addressed by evolutionary and chemical analysis. The 20 amino acids of the standard genetic code are not the only chemically possible amino acids, and dozens of others exist in biology in non-standard roles or as metabolic intermediates. The selection of exactly 20 appears to reflect a historical contingency rather than a chemical necessity, but the set that was selected shows evidence of having been chosen for its chemical diversity. It covers a broad range of chemical properties, including acidic and basic groups, polar and nonpolar side chains, and various sizes and shapes, suggesting that the selection of the genetic code's amino acids was not random, but was shaped by selection for proteins with broad functional potential. The expansion of the genetic code beyond 20 amino acids, which would require reassigning existing codons or adding new codon-anticodon pairs, is constrained by the universality of the existing code. Any change would affect every protein in the cell simultaneously, making beneficial expansion extremely difficult once the code is fixed. The philosophical question is large, but it can be held quietly. Does chemistry tend toward life when conditions allow, or was Earth extraordinarily lucky? Science does not yet know, but the answer is being approached through experiments, missions, genomes, ancient rocks, and models of other worlds. Wonder here is not separate from evidence. It is evidence followed patiently into one of the deepest unknowns. The theory of life's origin intersects with deep philosophical questions about the nature of life itself, what distinguishes living from non-living matter, and where exactly in the spectrum from complex chemistry to simple biology the threshold lies. There is no sharp chemical discontinuity between a very complex self-catalytic chemical system and the simplest conceivable living cell. The transition from chemistry to biology is more a matter of increasing complexity and the emergence of new properties than a sudden qualitative leap. The origin of life may therefore be better understood not as a single event, but as a process extended over millions of years, during which the distinctive features of life, metabolism, heredity, compartmentalization, evolution, gradually emerged and became integrated into the coherent, self-reproducing systems we recognize as living cells. The gradual nature of this transition makes it simultaneously more comprehensible, because it does not require an improbably sudden emergence of a fully functioning cell, and more difficult to study, because there may be no single moment that can be identified as the beginning of life. The body's connection to this story is direct. Every cell uses membranes, water, ions, carbon compounds, nucleic acids, amino acids, ribosomes, and energy gradients. These are not abstract textbook parts. They are ancient solutions still operating silently in the present. The origin of life is not only a remote event billions of years ago. It is the beginning of the processes that still make resting, breathing, healing, thinking, and sleeping possible. The wonder of the origin of life problem, and it is genuinely wonderful, is that it asks the most fundamental possible question about the universe's relationship to life. Whether life is a rare and precious accident in an otherwise indifferent cosmos, or whether the universe, through its chemistry and physics, is in some meaningful sense disposed toward the emergence of biology wherever the conditions allow. The scientific evidence does not yet provide a definitive answer to this question, but the richness of prebiotic chemistry, the abundance of organic molecules in space, the speed with which life appeared on Earth after stable oceans formed, and the extraordinary range of conditions under which life has been found to persist, all of this points toward a universe that is, at minimum, not hostile to life. Whether the universe is actively creative of life, whether biology is, in some deep physical sense, written into the chemistry of matter, remains an open question, and perhaps the most interesting open question in science. The early Earth returns in thought as a planet of magma, impacts, cooling crust, and first oceans, but now with more softness. The violence was real, yet it did not define the whole story. Between impacts, in protected pores, shallow waters, icy films, or mineral surfaces, chemistry could continue. Time did not guarantee life, but it offered countless chances for matter to become organized in new ways. The atoms in your body were forged in the cores of stars that lived and died before the solar system existed. They were assembled into organic molecules in the cloud of gas and dust that became the sun and its planets. They were incorporated into the early chemistry of the young Earth, and eventually, through processes that are still incompletely understood, organized themselves into the first living things. They have been cycling through the biosphere for 4 billion years, passing through billions of organisms, before arriving at their current configuration in you. You are the universe, organized into a form that can ask how the organizing began. The oldest evidence remains uncertain, but uncertainty itself can be informative. It tells us how delicate early traces are, how much geology can alter them, and why scientists compare structures, isotopes, minerals, and context before making claims. A claim about the first life is never just a claim about biology. It is also a claim about rock history, preservation, contamination, temperature, pressure, and alternative chemistry. That is, by any measure, quite a thing to be. Another way to understand the origin of life is through the word continuity. A living cell is not a statue. It is a pattern maintained through change. Molecules enter and leave. Bonds form and break. Proteins fold and degrade. Membranes repair themselves. Information is copied, damaged, and corrected. The living thing persists not because its material never changes, but because its organization is continually renewed. The first living systems may have been the first chemical organizations that could maintain continuity across time. This continuity would have been delicate at first. A protocell might lose its contents. A replicating molecule might break. A useful reaction might run out of material. A membrane might dissolve if conditions changed. But delicate does not mean impossible. Delicate things can become stable when conditions repeat. A shoreline that dries every day, a vent that flows for thousands of years, a mineral surface that keeps attracting the same kinds of molecules, or an ice channel that protects fragile chemistry can give delicate systems repeated chances. The origin of life is sometimes imagined as a search for the first spark, but sparks vanish quickly. A better image may be a candle protected from wind, or a small current that finds a channel. What matters is not only that a reaction happened, but that the products of the reaction remained available for the next step. Persistence is the quiet bridge between chemistry and biology. Without persistence, complexity washes away. With persistence, complexity can begin to accumulate. Modern life carries signs of deep ancestry in places that are easy to overlook. ATP is used everywhere in cells. Ribosomes are shared by all cellular life. The genetic code is nearly universal. Cell membranes separate inside from outside. Proton gradients power metabolism. These features are so familiar to biology that they can feel ordinary, but they are not ordinary at all. They are ancient solutions that became so useful that life kept them through billions of years of change. One of the most humbling facts is that Luca was already laid in the story. By the time the ancestors of all living cells shared a common population, much had already happened. The genetic code existed. Translation existed. Metabolism existed. Some kind of membrane biology existed. The path from prebiotic chemistry to Luca may have contained whole vanished worlds of molecular evolution. We do not see them directly, but their absence creates the depth behind the earliest common ancestor. This hidden pre-Luca world may have included systems that would seem strange beside modern cells. Some may have used RNA more heavily. Some may have had leakier membranes. Some may have exchanged genes constantly. Some may have depended directly on geochemical settings. Some may have lacked clean species boundaries. The early biosphere may have been experimental in a way later life no longer is. Stabilization came gradually as successful systems became harder to displace. There is a quiet comfort in the fact that life did not need to be perfect to begin. It needed to be good enough to continue. The first copying systems could make errors. The first membranes could leak. The first metabolisms could be inefficient. The first cells could be vulnerable. Evolution does not require perfection at the starting line. It requires heritable differences and consequences. From that modest beginning, natural selection can become powerful. The same principle applies to knowledge. Science does not need a perfect reconstruction of the first cell before it can understand pieces of the transition. One experiment can show how vesicles form. Another can show how nucleotides might arise. Another can show how our Na can catalyze reactions. Another can show how minerals concentrate molecules. Each piece is incomplete, but incompleteness is not emptiness. It is a map with blank regions slowly being filled. The origin of life also reminds us that Earth itself is not passive. The planet provides gradients, cycles, boundaries, and materials. Day and night are cycles. Wetting and drying are cycles. Freezing and thawing are cycles. Volcanic activity brings new chemistry. Impacts reset and enrich environments. Oceans dissolve and transport compounds. Minerals concentrate and catalyze. A planet can be chemically creative without being alive. Life may be what happened when that creativity found a way to remember itself. And once memory appeared, the planet changed. Biology altered minerals, air, oceans, and climate. Microbes helped shape sediments. Photosynthesis changed atmospheric chemistry. Life became a geological force. The origin of life was therefore not only the appearance of organisms on Earth. It was the beginning of Earth becoming a living planet, a world whose surface chemistry would forever be shaped by cells. The body resting here now belongs to that same planetary continuity. Its cells use ATP, membranes, ribosomes, amino acids, and nucleic acids. Its carbon has traveled through air, food, and life. Its oxygen once belonged to water, rock, and atmosphere. Its phosphorus and nitrogen entered through cycles older than civilization. Nothing in the body is isolated from Earth's history. The origin of life is not only far away in time, it is present in the molecular habits still keeping the body alive. So the subject can become peaceful instead of overwhelming. It says that the deepest beginnings are often small. A membrane, a molecule, a gradient, a pore, a droplet, a strand, a cycle. The scale of the result is enormous, but the steps may have been intimate. Life's beginning may have happened in spaces smaller than a grain of sand, yet it eventually filled oceans, soils, skies, and imaginations. When all of these ideas are held together, the origin of life begins to feel less like a single spark and more like a long dawn. A young planet cooled. Water gathered. Rocks reacted. Lightning flashed. Meteorites fell. Vents released hydrogen into dark ocean water. Ponds filled and dried. Minerals held molecules close. Fatty acids made fragile boundaries. RNA-like molecules explored sequences. Some systems lasted a little longer than others. Some copied. Some changed. Some became lineages. From that beginning, the story widened. Microbes transformed the oceans. Photosynthesis changed the air. Cells merged. Eukaryotes appeared. Multicellular life came much later. Animals came later still. Minds came very late. And now a mind can rest in the thought that it is made of matter old enough to remember stars, organized by life old enough to remember the early Earth, asking how the organizing began. The question does not need to be finished tonight. It can remain open, like a dark ocean under a young sky. Each experiment adds a small lamp. Each ancient mineral adds a hint. Each living cell carries a trace. The beginning is not fully visible, but it is not absent. It is present in every breath, every heartbeat, every green leaf, every microbe dividing unseen. Life began somewhere in the deep past, and it has not stopped continuing since. Every cell division is a continuation of that original transition. Every seed, every colony of bacteria, every sprouting plant repeats the ancient theme of matter organizing itself into persistence, not from nothing and not outside nature, but through chemistry made patient by time. The universe is not required to explain itself all at once. Neither is life. Tonight it is enough to let the scale soften. The atoms are old. The oceans were ancient before animals appeared. The first cells were small beyond sight, yet everything alive carries their inheritance. The path from chemistry to biology remains partly hidden, but the fact that the path existed is written everywhere, in cells, in genes, in metabolism, in the air, in the forests, and in the quiet body resting here now. There is another quiet way to think about the first boundary. A membrane does not have to be intelligent to matter. It only has to keep some molecules near one another longer than chance would allow. Inside a vesicle, a useful strand and a useful small molecule can meet again. Outside, in open water, they may separate forever. The boundary changes probability. It gives chemistry a room, and rooms allow relationships to persist. The earliest membranes may also have been leaky, and that may have helped. A perfectly sealed protocell would trap its contents, but also starve. A very leaky one would lose everything. Between those extremes is a useful middle state. Small molecules can enter, waste can leave, and larger useful polymers remain inside. Modern cells control this movement with exquisite machinery, but early protocells may have depended on the natural properties of simple fatty acid membranes. Growth and division could have begun without any modern machinery. A vesicle that absorbs more fatty acids can increase its surface area. If it becomes long, unstable, or physically disturbed, it can split into smaller vesicles. This is not the same as modern cell division, but it creates a primitive rhythm: grow, distort, divide, continue. If the contents inside influence that rhythm, then chemistry inside the boundary begins to matter to the future of the boundary itself. The first selection may have been almost invisible. A vesicle containing an RNA-like polymer might swell differently from an empty vesicle. A molecule that helped make membrane material might indirectly help its own container. A protocell that retained useful compounds might grow a little faster. None of this requires intention. It requires only differences that affect persistence. Evolution begins quietly when differences begin to matter. Replication also did not need to begin with the elegance of modern DNA copying. A short strand can guide the formation of a complementary strand if matching building blocks line up along it. The chemistry is difficult, and many details remain unresolved, but the principle is clear. Sequence can influence sequence. Once one molecule helps shape the order of another, information has begun to enter chemistry in a new way. Errors would have been unavoidable. Early copying systems were probably messy, slow, incomplete, and fragile. But errors are not only failures. Some errors are variations. A molecule that copies with no variation cannot explore. A molecule that copies with too much error loses itself. The earliest evolving systems had to find a balance between remembering and changing. That same balance still runs through every genome today. The idea of an error threshold is important here. If a replicating molecule is too long and copying is too inaccurate, the information cannot be maintained. This may have limited the size of the earliest genomes. Small systems could survive with higher error rates. Larger systems needed better copying. The evolution of improved replication may therefore have opened the door to more information, more functions, and more elaborate protocells. Cooperation may have appeared before cells were fully alive. One molecule might catalyze a reaction that helps another. A second molecule might stabilize the first. A third might help gather a building block. Networks like this can be fragile because parasites can exploit them, but they also offer more possibilities than lonely molecules. Life may have begun not with one heroic molecule, but with chemical relationships that reinforced one another. Parasites, in a molecular sense, may have been present very early. If a short RNA-like strand could be copied faster than a useful longer one while using the same resources, it could spread without helping the whole system. This would create pressure for compartments, because a compartment can link the success of molecules inside it. Helpful molecules benefit their own container. Pure parasites can damage the container that carries them. Boundaries turn chemistry into ecology. Ecology may therefore be older than cells in the modern sense. Even a set of protocells could compete for fatty acids, nucleotides, minerals, light, or energy-rich molecules. Some could exchange contents. Some could merge. Some could break. Some could become more stable. When many protocells exist together, their environment is not only rock and water, it is also one another. The origin of life may have included community before species. Temperature would have shaped everything. Heat speeds reactions, but it can also destroy fragile molecules. Cold preserves, but it can slow chemistry. Alternating temperatures may have been useful, allowing some reactions to occur during warm phases and some preservation during cooler phases. A planet with day and night, seasons, volcanic settings, ice, and water circulation offers many thermal patterns. Chemistry could sample them all. Ultraviolet light was more intense on the early Earth because there was no oxygen-rich atmosphere and no protective ozone layer like today's. Ultraviolet radiation can damage delicate molecules, but it can also drive important photochemistry. This double role is typical of origin of life research. A condition can be both dangerous and useful. The same light that breaks some bonds can help form others. The early Earth was not gentle in a simple way. It was chemically active. Impacts also had two phases. Large impacts could heat oceans, disturb crust, and destroy fragile systems near the surface. Smaller impacts and late accreted material could deliver metals, organics, and reducing power. Impact-generated atmospheres may have created temporary chemical windows, producing compounds useful for prebiotic chemistry. The origin of life may have depended on a planet that was dangerous but not hopeless, restless but not sterile forever. The faint young sun adds another layer. Early in Earth's history, the sun was dimmer than it is now, yet geological evidence suggests liquid water existed. This faint young sun problem has several possible answers, involving greenhouse gases, atmospheric pressure, clouds, and changing planetary conditions. For the origin of life, the important point is that early Earth was not simply today's Earth with fewer organisms. It was a different climate system with different chemical possibilities. Oceans themselves were chemical worlds. Dissolved iron, carbon dioxide, salts, volcanic gases, and minerals would have shaped what reactions were possible. Before oxygen rose, iron could remain more soluble in seawater. Later, oxygen helped form vast banded iron formations as iron oxidized and settled. The chemistry of water changed as life changed the planet. The first life emerged in an ocean that no modern swimmer has ever touched. The oldest living lineages today are not windows that show the first life unchanged. Bacteria and archaea have also evolved for billions of years. Hyperthermophiles, methanogens, and other deeply branching organisms can offer clues about ancient metabolisms, but they are modern survivors, not preserved originals. This distinction matters. Evolution never paused. Even organisms in extreme settings are not fossils in the simple sense. They are living branches with their own long histories. Methanogenesis, acetogenesis, sulfur metabolism, and other ancient-looking pathways attract attention because they are deeply rooted and geochemically simple in some respects. They use small molecules such as hydrogen, carbon dioxide, sulfur compounds, and metals. Such metabolisms feel close to the chemistry of rocks and vents. Whether they represent something very near the beginning or later biological refinements is debated, but they show how intimately early life may have been tied to geochemistry. The citric acid cycle, known in modern cells as a central part of metabolism, also has origin of life echoes. Some researchers study partial reverse cycles or related networks that could fix carbon dioxide into organic molecules. Modern metabolism may contain traces of older chemical pathways, transformed by enzymes over time. It is possible that before enzymes were precise, minerals and simple catalysts guided rougher versions of reactions that life later refined. The emergence of proteins changed everything. RNA can catalyze reactions, but proteins offer a wider range of chemical possibilities because amino acids have diverse side chains. Once translation linked RNA sequences to amino acid sequences, natural selection could explore protein space. Better catalysts could arise. Better catalysts could help replication, metabolism, and membranes. Translation was therefore not just one invention among many. It connected information to chemistry with extraordinary power. Yet translation itself is difficult to explain. It requires RNA adapters, ribosomal machinery, a code connecting codons to amino acids, and systems for attaching amino acids to the correct adapters. This complexity suggests a long period of partial solutions. Perhaps short peptides first helped RNA. Perhaps RNA molecules bound certain amino acids directly. Perhaps early codes were simpler and expanded over time. The modern system looks finished, but its origin was probably gradual. DNA's later rise also makes sense in this gradual view. RNA is versatile but chemically vulnerable. DNA is more stable and easier to repair in certain ways. As life became more complex and genomes became larger, a more durable archive would be favored. The shift from RNA-centered heredity to DNA genomes may have been one of life's great stabilizing transitions. It allowed information to become more securely stored while our RNA and proteins performed faster work. Once DNA, RNA, and proteins formed a partnership, biology had a powerful division of labor. DNA could store, RNA could transfer, regulate, and catalyze in special places. Proteins could catalyze an immense variety of reactions. Membranes could create controlled spaces. Energy systems could power the whole arrangement. This is the architecture that every living cell inherits in modified form. It is so familiar that its strangeness can be easy to miss. The first fossils of life, whatever exact examples are accepted, are much later than the first chemistry that led to life. Fossils preserve organisms or their activity after living systems were already established. The molecular beginning itself may be nearly invisible in rocks. This means laboratory chemistry and comparative biology are not optional, they are necessary. Rocks tell one part of the story, living cells tell another. Experiments help bridge the darkness between them. Ancient carbon isotope evidence is powerful because life tends to prefer lighter carbon isotopes during metabolism. This preference can leave a measurable signature in ancient carbon. But non-biological processes can sometimes complicate interpretation, especially in rocks that have been altered. The best scientific claims are careful because they know ancient evidence is delicate. Certainty grows when multiple lines of evidence converge: shape, context, chemistry, isotopes, and geological setting. Stromatolites are among the most evocative ancient signs of microbial life. They are layered structures often built by microbial mats that trap and bind sediments. Modern stromatolites still exist in places such as Shark Bay, though ancient examples require careful interpretation. Their layered shapes remind us that for most of Earth's history, life did not announce itself with animals. It built films, mats, textures, and chemical changes. It was quiet, persistent, and planetary. The long microbial age is relaxing because it removes the need for hurry. For billions of years, life was mostly single-celled. During that time, it was not stagnant. It invented metabolisms, reshaped environments, exchanged genes, and prepared the chemical foundations for later complexity. The absence of animals was not an absence of progress. It was a deep biochemical history taking place at scales too small for eyes. Another overlooked part of origins research is time between events. A molecule does not need to survive forever if the environment keeps making it again. A fragile compound can matter if it is continually replenished by photochemistry, impacts, vents, or mineral reactions. Early chemistry may have depended less on permanent stability and more on repeated production. A stream can stay full even though each drop moves on. This repeated production helps explain why local environments matter. If a pond dries once, it may produce a small set of polymers. If it dries thousands of times, across many seasons and many pools, rare products have more chances to appear. If an impact-generated atmosphere makes useful molecules for a few million years, that is not a moment to a planet. It is a long chemical weather pattern. The first successful lineage may also have benefited from environmental selection before biological selection fully existed. Molecules that survived ultraviolet light, heat, acidity, or mineral binding would become more common in certain places. Vesicles stable in one kind of water would persist there better than others. Before genes were selected, chemistry itself was filtered by the planet. Earth acted as a sieve, keeping some possibilities and washing others away. There is a quiet relationship between damage and creativity. Ultraviolet light can damage RNA, but it can also help build nucleotide precursors. Heat can break molecules, but it can also speed reactions. Impacts can sterilize a region, but they can also deliver metals and create temporary atmospheres. Oxygen later became dangerous to anaerobic life, then became fuel for complex metabolism. Life's history repeatedly turns hazards into resources. The origin of life may have required not a perfectly comfortable planet, but a planet with enough contrast. Hot and cold, wet and dry, acidic and alkaline, oxidizing and reducing, mineral and organic, light and dark. Gradients store possibility. A perfectly uniform environment is chemically sleepy in the wrong way. Differences allow flow, and flow allows work. Life is one of the ways the universe uses differences before they fade. This is why equilibrium is the opposite of life. At equilibrium, there is no usable gradient, no net flow, no chemical reason for one direction over another. Living systems remain far from equilibrium by constantly exchanging energy and matter with their surroundings. The first living systems must have found a way to sit in a flow without being destroyed by it. They were not closed boxes. They were open patterns. A protocell, then, can be imagined as a small open pattern. Building block center. Some reactions happen. Some products remain. The boundary grows. The contents influence the boundary. The boundary protects the contents. If division occurs, part of the pattern continues elsewhere. This is not yet a modern cell, but it has the outline of inheritance. It has a before and an after. The earliest evolution may have been shaped by drift as well as selection. In small protocell populations, chance matters. A useful molecule can be lost. A less useful one can become common by accident. Later, stronger selection may refine systems, but beginnings can be messy. This does not weaken the story. It makes it more realistic. Life did not emerge from a perfectly ordered process. It emerged from chemistry, chance, constraint, and time. Laboratory origin of life experiments often focus on one piece at a time because the whole problem is too large for one flask. One setup explores nucleotide formation. Another explores vesicle growth. Another explores mineral catalysis. Another explores RNA evolution. The challenge is to connect the pieces without pretending the connections are already proven. The field advances by building bridges between partial successes. Some of the most exciting work now asks how these pieces interact. Can a membrane survive conditions that make RNA building blocks? Can RNA replicate inside a vesicle? Can protocells grow while carrying genetic polymers? Can primitive metabolic reactions feed the production of membrane material or activated nucleotides? These questions matter because life is integration. A list of parts is not yet a living system. The beginning of life may also have involved molecules that modern life no longer uses in the same way. Pre-RNA worlds are possible, involving simpler genetic polymers that might have preceded RNA. Alternative backbones, related nucleic acids, or mixed systems may have existed and vanished. Modern biology may preserve only the winners. The early chemical world could have been more diverse than the one that survived. This survivor bias is important. We know the path that led to current life because current life is here. We do not know how many paths ended. Early Earth may have produced many promising systems that failed because conditions changed, resources vanished, parasites overwhelmed them, or more efficient systems replaced them. The origin of life may have been full of extinct beginnings, none of them visible now except through principles discovered in experiments. Even the word first can be misleading. There may have been a first self-copying molecule, a first useful membrane, a first protocell lineage, a first genetic code, a first DNA genome, and a first cell in a more modern sense. These firsts may not have happened at the same time. Life's origin may be a sequence of thresholds rather than a single threshold. The question becomes not only when life began, but when each feature of life became linked to the others. For a calm mind, this layered beginning is easier to hold. It does not ask one molecule to carry the entire mystery. It lets each feature arrive gradually. Energy before ATP, compartments before modern membranes, catalysis before enzymes, heredity before DNA, selection before species, ecology before ecosystems with names. The great complexity of life becomes a set of smaller transitions, each still difficult, but no longer impossible to imagine. And the deepest relaxation may come from knowing that life is not separate from the universe that made it. The same laws that move planets and shape stars also govern bonds, water, membranes, and metabolism. Life is unusual, but it is not outside nature. It is nature arranged into self-maintaining, self-copying patterns. In that thought, the origin of life becomes not a rupture in the world, but one of the world's most delicate continuities. The origin of life also changes the meaning of ordinary water. Water is not only a background liquid. It folds some molecules and unfolds others. It carries ions. It allows diffusion. It participates in reactions. It helps membranes form because oily tails avoid it while polar heads face it. The same water that can make polymer formation difficult can also make compartments possible. This double nature is part of why the problem is rich rather than simple. Salt matters too. Different ions can stabilize or destabilize fatty acid vesicles, RNA structures, and mineral surfaces. Magnesium can help some RNA chemistry, but can also damage fatty acid membranes under certain conditions. This kind of conflict is important. A condition useful for one part of early life may harm another. The first successful systems had to find compatible combinations or environments where conditions changed in the right order. Order of events may have been as important as ingredients. A pool might first concentrate small molecules, then dry enough to join them, then refilled gently enough to let membranes form, then cool enough to preserve what was made. In another setting, a vent might provide energy first, while mineral pores provided boundaries before lipid membranes existed. Origins research often asks not only what was present, but what happened before what. The early Earth had no clean separation between chemistry and geology. A molecule could form in the atmosphere, rain into a pond, bind to a mineral, be heated by volcanic ground, dry into a film, wash into a stream, enter the ocean, and meet vent chemistry later. The planet was connected. This makes one location stories too simple, but it also makes the possibilities richer. Ingredients could travel between laboratories made by nature. There is a comforting humility in this. No single hypothesis has to carry the whole origin alone. The pond can explain concentration. The vent can explain energy. The meteorite can explain delivery. The mineral can explain catalysis. The vesicle can explain boundaries. The RNA molecule can explain early heredity. A realistic beginning may have borrowed from all of these, not as a tidy plan, but as a sequence of opportunities. Finally, the question of life's origin reminds us that the present is layered with the past. Every breath depends on oxygen released first by ancient microbes. Every thought depends on cells powered by mitochondria descended from bacteria. Every gene belongs to a coding system older than animals. Every protein is built by ribosomes whose catalytic core is our RNA. The beginning has not disappeared. It is still working quietly inside life now. A small molecule on early Earth had no destiny by itself. It could be destroyed by heat, diluted by water, trapped on a surface, activated by light, or carried into a new setting. The origin of life depends on the fact that some molecules did not remain alone. They became part of sequences, membranes, cycles, and gradients. A molecule became meaningful when it helped the system continue. That is why the earliest life may have been less a thing than a relationship. A replicator related to its building blocks. A membrane related to its contents. A gradient related to the reactions it powered. A mineral surface related to the molecules it held. Biology began when these relationships could be inherited in some form, so that the success of one moment influenced the next. The most relaxing part of this is that complexity did not need to know where it was going. It only needed to preserve what worked locally. A strand that copied slightly better, a compartment that lasted slightly longer, a reaction that produced a useful building block. Each could matter without any plan. Across deep time, such small mattering became the living Earth. There is another quiet way to understand the earliest Earth, as a place where stability and instability overlapped. A perfectly violent planet would have erased every fragile experiment before it could continue. A perfectly still planet might not have driven enough chemistry forward. The young Earth seems to have offered something in between. Heat, impacts, water, rock, gases, minerals, and changing environments, but also protected pockets where chemistry could persist long enough to become interesting. Origin of life research often lives in that middle space. It looks for settings active enough to make molecules, but gentle enough to let them survive. The word ingredient can be misleading if it makes the beginning sound like a recipe. Carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, iron, nickel, and magnesium were not placed neatly into a bowl. They moved through atmosphere, ocean, crust, and falling debris. They changed oxidation state. They dissolved, precipitated, evaporated, froze, and reacted. A building block of life is not alive by itself. It becomes relevant only when the surrounding environment gives it concentration, timing, protection, and a path into larger organization. Concentration is one of the quiet heroes of the story. The open ocean is vast, and vastness dilutes. Dilution can make fragile reactions unlikely, because molecules that need to meet are carried too far apart. This is why ponds, films on rocks, pores in minerals, ice channels, sea spray droplets, and vent chimneys matter so much. They make small places inside a large planet. They bring molecules close enough to interact. Before a cell had a membrane, the planet itself may have offered temporary rooms. Cycles matter as much as places. Wetting and drying can join molecules that would not easily link in continuous water. Freezing can push solutes into narrow, salty channels. Heating can drive reactions, while cooling can preserve products. Daylight can damage some molecules and activate others. A cycle does not need to know what it is doing. It only repeats. With every repetition, chemistry may move a little farther from simple mixture and a little closer to organized persistence. This is why surface pools and deep vents are not only rivals in the story. They may represent different gifts. A surface pool can concentrate molecules through evaporation and expose them to ultraviolet light. A vent can provide continuous chemical energy, mineral compartments, and redox gradients. A shoreline can mix atmosphere, water, and rock. Ice can protect and concentrate. The beginning may have borrowed from more than one setting, with chemistry moving through landscapes rather than remaining in a single birthplace. RNA receives so much attention because it offers a bridge between information and action. Yet RNA is also fragile, complicated, and difficult to make under many ordinary conditions. That difficulty is not a reason to discard the idea. It is the reason the research is active. Chemists test pathways that make nucleotides through shared precursor chemistry. They test minerals and wet-dry cycles that encourage polymerization. They test shorter RNA-like molecules and mixed systems that may have existed before modern RNA became dominant. Some researchers therefore imagine not a pure RNA world, but a messy world where RNA, like strands, short peptides, lipids, minerals, and simple metabolites helped one another. Peptides could stabilize RNA. RNA could bind amino acids. Small molecules could feed reaction networks. Membranes could keep useful combinations from drifting away. This cooperative picture is less tidy than a single molecule solution, but early Earth was not tidy. It may have been rich precisely because many partial systems overlapped. Metabolism first ideas offer another restful perspective. Instead of beginning with a molecule that copies itself, they begin with networks of reactions that transform carbon and energy. Mineral surfaces, especially iron-sulfur minerals, could have guided some of those transformations. Hydrogen from water-rock reactions could have reduced carbon dioxide into simpler organic compounds. In this view, life begins as flow before it becomes inheritance. The first step is not a gene, but a self-feeding chemistry that makes the next reaction easier. The challenge is that metabolism without heredity cannot evolve in the full Darwinian sense. It can persist, but it cannot easily accumulate improvements across generations. A copying molecule without metabolism has the opposite problem: information without a reliable energy and material supply. The origin of life may therefore have required these two streams to meet. When a reaction network helped a replicating system, and the replicating system helped preserve the network, chemistry began to acquire memory. Membranes turn memory into a place. A molecule floating freely in water can copy, break, or drift away, but its success may not help a larger system. Inside a vesicle, the story changes. If the molecule helps the vesicle grow, divide, or gather building blocks, then the boundary and the contents begin to share a fate. This shared fate is one of the earliest shadows of individuality. The protocell is not yet a modern cell, but it is no longer just a soup. Early membranes may have been very unlike modern membranes. Modern cell membranes use carefully constructed phospholipids and proteins. Prebiotic membranes may have used simpler fatty acids or mixed amphiphiles that assembled more easily. They may have been leaky, and that leakiness could have been useful. A perfectly sealed protocell might starve before it had transport proteins. A gently leaky vesicle could let small nutrients enter while keeping larger informational molecules inside. Imperfection may have been part of the path. Energy currencies also may have had earlier forms. Modern ATP is elegant, but it belongs to an advanced cellular world. Before ATP dominated, simpler activated phosphate compounds, or thioesters, may have helped drive reactions. Natural proton gradients at alkaline vents may have offered another kind of usable energy. The question is not which modern system appeared fully formed, but which primitive forms of energy handling were good enough to support growth, copying, and selection. Phosphorus is a small but important difficulty in this story. Life needs phosphate for nucleic acids, ATP, and membranes, but phosphate minerals can be poorly soluble under many conditions. Researchers therefore study special environments where phosphorus could become more available: volcanic settings, impact chemistry, evaporating pools, mineral surfaces, or reduced phosphorus compounds delivered by meteorites. The problem is not that phosphorus was absent, but that early chemistry had to find usable pathways for it. Chirality adds another quiet mystery. Life uses one-handedness for amino acids and another for sugars, producing polymers that fold reliably. A mixture of both handednesses can disrupt structure. The first bias may have been small, perhaps produced by chance, polarized light, mineral surfaces, or extraterrestrial chemistry. But a small bias is not enough by itself. It must be amplified and preserved. The origin of homochirality is therefore a story about tiny asymmetries becoming biological consistency. The fossil record of the earliest life is difficult because deep time is not gentle with evidence. Rocks are heated, squeezed, altered, melted, eroded, and recycled. A structure that looks biological may be mineral. A chemical signature that looks biological may have non-biological explanations. This is why careful language matters. The oldest evidence for life is not a single dramatic fossil that ends the discussion. It is a set of traces, each weighed against geology, chemistry, and preservation. Luca is often emotionally tempting to treat as the first life, but it was almost certainly not that. Luca was the last common ancestor of life that survived into the present, not the first chemical system that crossed the living threshold. Many earlier lineages could have existed and disappeared without descendants. Some may have contributed genes or chemistry to the line that survived. The visible tree of life may begin after a long invisible forest had already grown and vanished. That thought makes the story deeper rather than weaker. The fact that we cannot see every early branch does not mean nothing happened. It means the surviving record is selective. Every living cell today is a witness, but a witness that has been rewritten by billions of years of evolution. The earliest chapters are not preserved as pages. They are preserved as molecular habits, ribosomes, code, membranes, gradients, cofactors, and the strange universality of cellular life. Cofactors are another clue that early biochemistry may have grown from geochemistry. Many enzymes use metal clusters or small organic helpers to move electrons and rearrange molecules. Iron, sulfur, nickel, molybdenum, and other elements appear at the heart of ancient metabolic reactions. These metals were not invented by life. They were part of rocks and water before life began. Biology may have learned to hold and refine chemical powers that minerals already displayed in rougher form. Ocean worlds elsewhere make the origin of life feel less lonely. Europa and Enceladus appear to have liquid water beneath ice. Enceladus sends plumes into space, carrying material from its hidden ocean. Mars once had rivers, lakes, and habitable environments on its surface. None of this proves that life exists or existed there. It only means that the questions asked about early Earth now echo across the solar system. Water, rock, chemistry, and energy are not unique ideas found only at home. Biosignatures require the same caution as ancient fossils. Oxygen can be produced by life, but also by some non-biological processes under certain conditions. Methane can be biological, geological, or produced by water-rock reactions. A single gas is rarely enough. Scientists look for patterns, disequilibrium, context, replenishment, planetary environment, and consistency across observations. The search for life elsewhere is therefore not a hunt for one magic sign, but a patient reading of whole worlds. This patience makes the origin of life a good subject for a quiet night. It does not rush towards certainty. It does not pretend that every gap is already filled. It lets mystery remain where mystery is honest. At the same time, it does not surrender to mystery. It keeps testing chemistry, reading rocks, comparing genomes, and building protocells in the laboratory. The beauty is in the balance. Humility without emptiness, curiosity without noise. There is also tenderness in the scale of the transition. The first systems were probably microscopic, fragile, and unimpressive to any eye. No forest rose from the first protocell. No animal moved. No flower opened. Yet the whole later biosphere depended on the ability of small things to continue. This is one of the deepest lessons in natural history. The greatest transformations can begin below visibility, in processes too small and slow to feel dramatic while they are happening. And that is why the origin of life can feel relaxing rather than frightening. It places the body inside a long continuity of matter-learning organization. The cells resting now are not separate from ancient chemistry. They still use membranes to hold an inside. They still use gradients to move energy. They still use nucleic acids to carry instructions and ribosomes to turn those instructions into proteins. The beginning is distant, but its solutions are still quietly active here. There is a useful difference between making building blocks and making life. Amino acids, sugars, nucleobases, and fatty acids are important, but they are still only parts. The deeper question is how parts became organized into a system that could continue. A pile of letters is not a sentence. A pile of molecules is not a cell. Organization, cycling, and inheritance are what make the story cross from chemistry into biology. Laboratory work is powerful because it separates one piece of the mystery at a time. One experiment may make nucleotides. Another may grow vesicles. Another may evolve ribozymes. Another may test mineral surfaces. None of these experiments recreates the whole beginning, and that is fine. Science often approaches a vast unknown by building reliable islands of knowledge, then asking how the islands might connect. One danger in origin of life storytelling is making the first cell too modern. The earliest systems probably did not have polished enzymes, perfect membranes, accurate genomes, or neat metabolisms. They may have been noisy, leaky, chemically mixed, and easily lost. That roughness is not a problem. It makes the beginning more plausible. Evolution can improve systems once there is something imperfect but heritable enough for selection to act on. Another danger is imagining that every molecule had to be pure. Modern cells use carefully selected molecules, but prebiotic Earth may have been chemically messy. Mixed membranes, mixed polymers, side products, damaged molecules, and alternate pathways may have been everywhere. Some mixtures would fail, some would interfere, but some mixtures could stabilize each other or open a new path. Life may have grown not from perfect purity, but from useful mess. The idea of a threshold is helpful because it avoids a single magical instant. There may not have been one first second when life began. Instead, systems may have become more lifelike over time, first concentrating, then cycling, then copying, then enclosing, then selecting, then evolving more clearly. The threshold could have been crossed gradually, with chemistry becoming biological by degrees. Every modern cell is a kind of archive, but not a simple archive. Its genes have changed. Its proteins have changed. Its membranes have changed. Yet some themes remain extremely old. Information must be copied. Energy must be handled. Boundaries must be maintained. Materials must be imported and waste removed. Even the simplest cells are solutions to these ancient demands. The origin of life asks how those demands first became linked. The role of water is beautifully double. Water makes life possible by dissolving and transporting molecules, but it can also make some bonds harder to form. Water encourages membranes because oily parts avoid it, but water can dilute reactants until they rarely meet. This double nature is why origin scenarios often include cycles or surfaces. Life did not begin in abstract water alone. It began, if it began here, in water interacting with place. The role of time is just as double. Time can destroy fragile molecules through breakdown, dilution, and impacts. But time also allows rare events to occur, cycles to repeat, and successful arrangements to be tested again. Early Earth offered millions of years of chemical opportunity. That does not make life inevitable, but it changes the scale of probability. A difficult step repeated across countless environments may become less lonely. A calm mind can hold all these models together without forcing them to agree too soon. Our NA world, metabolism first, lipid world, mineral catalysis, vents, ponds, ice, meteorites. Each gives a partial answer. The most realistic picture may be a network of partial answers. The beginning of life may have been plural in its chemistry before the surviving lineage became singular in ancestry. In the end, the origin of life remains both a scientific problem and a quiet form of perspective. It tells us that the boundary between the living and non-living world is not a wall of substance. Living things are made of ordinary atoms. The difference is organization, flow, information, and continuity. Matter did not stop being matter when it became alive. It became matter arranged in a way that could remember and renew itself. That thought can settle gently. The body does not need to solve the origin tonight. It can rest inside the fact that the same ancient principles are still working. Membranes holding cells together, gradients powering reactions, molecules carrying information, proteins doing quiet work in the dark. The beginning is hidden in deep time, but the pattern that began there is close, present, and breathing. And perhaps the most peaceful conclusion is that uncertainty does not make the story smaller. It keeps the story open. New experiments may change the details. New missions may find unexpected chemistry on other worlds. New genomic work may revise Luca again, but the central wonder will remain. On a young Earth, ordinary matter entered a kind of continuity that could evolve, diversify, and eventually wonder about itself. And because the beginning is still partly hidden, the mind does not have to force it into a single shape. It can simply rest with the evidence. Ancient rocks, living cells, laboratory chemistry, meteorites, oceans, minerals, membranes, and time all pointing toward the same quiet threshold. Sleep well. Good night.